And my lab has moved to UC San Diego last week. And we are interested in epigenetic control of gene regulation. So the epigenome controls gene regulation in many ways, including, for example, controlling where the chromatin is more open, and also individual transcription factors could interact with particular kinds of epigenetic modifications. Remain not completely solved are where are the regulatory sequences and what are their exact regulatory functions. Among many difficulties, one of them is that there are certain numbers of transcription factors in the human genome times certain numbers of potential epigenetic modifications. There is a big number of potential interactions. For us to look through every one of these interactions seems to be a challenge. Our original inspiration came from comparative genomics, where since the conservation or the rate of change of specific parts of the human genome could be used to infer either negative or positive functions, could we also compare the genome in the context of the epigenome? That was the original thought. Not like the beautiful work that have already been done for the genome evolution aspect. How the epigenome evolved has not as extensively explored. So we wanted to build some, or to start to explore some basic properties underlying the epigenome evolution. We wanted to do that in the context of genome evolution, so that we wanted to look at the co-evolutionary properties between the epigenome and the genome, between the genome and transcriptome, as well as between the epigenome and the transcriptome. For now, I would like to use 1, 2, and 3 to denote these types of co-evolutionary properties that we started to look at. To do this, we wanted to choose some biological processes that are thought to be conserved across mammalian systems. One of those systems was early development. This preimplantation development process was thought to be a quite conserved process. So we started, a few years back, we started to assay transcriptome at multiple stages during this early developmental process in 3 mammalian systems. After clustering the gene expression data, we started to find gene clusters where the response of the transcription appears to be conserved across species in this process. It's not true for every cluster. As you can see, there are some clusters which appear to be not completely conserved across mammalian species. The question was that what drives such a conservation of the transcription response, or what drives the divergence of such response? And to look at this, we wanted to first look at the genome changes. We picked out one gene. This is MTF2, is one of the polycon group genes, which appear to have a divergent expression pattern between humans, cows, and mice. The mice seems to be reverse to the behavior of humans and cows. So we wanted to utilize transcription factor binding data generated by Huck Huang's group in the embryonic stem cells. So what we are looking at is the genome neighborhood of this particular gene assayed at about this time point. And you can see this gene goes this way. Some transcription factors bind to the upstream and intron of this gene, which might be transcription activators that are responsible for the upregulation of this gene in mouse only. We're looking at the ChIP-seq data in the mouse genome at this moment. Whereas the downstream binding site appears to be not conserved across species, the upstream binding site appears to be conserved across species, which was a surprise because we only saw the transcription factor binding in the mouse, which corresponded to the mouse-specific activation. How come that a conserved binding site didn't generate binding in the human genome? So it turned out that although the whole piece of sequence appeared to be conserved, there was a single nucleotide difference between the rodents and the other mammals. The other mammals all had a T, whereas this single base pair change in the rodents seems to make this piece of sequence accordant to the predicted motif that LANOG binds onto. And thus, we started to hypothesize that maybe there was a single base pair mutation that gave rise to a new binding site. If this theory was true, we could be able to mutate this single base back to the ancestral version and abolish the binding, which appeared to be the case in vitro. What happened to the downstream sequence was that there was a transposable element, a family of transposable elements that were specific to the rodents, which seemed to carry the motifs of these two proteins. And those motifs seems to be perfectly superimposed onto the minimal peaks, minimal overlap of ChIP-seq data of those two proteins. Now that we start to think maybe the jumping of transposable elements together with single base pair mutations could generate new binding sites and thus rewire the transcriptome. And thus, this gene becomes downstream to those upstream transcription factors. Altogether, these types of sequences changes, when we accumulate them, we find that we started to explain about 10% of the interspecies expression differences in the early development of those species. 90% of the divergence we couldn't explain at that time. That led us to think about, how about the epigenome? Can we add the epigenetic information in? To add epigenome into this interspecies comparison, we first need the data. We first need, we started to generate a 3-species epigenome map using pluripotent stem cells, because those cells, we sort of can somewhat match their cellular property. A number of published ChIP-seq data were put into this study, and we put in about 15 samples from 2 species, and we supplemented about 30 samples in different epigenetic modification and some transcription factors to make a 3-genome epigenome map, 3-species epigenome map. And this map looks like the following. Now that I'm looking at some orthologous regions across 3 species, the same color denote the orthologous sequences. And now that onto these orthologous regions, I have the intensity map of multiple epigenetic modifications. So now that I can start to investigate this data. Some of the early questions that we asked was whether local sequence conservation or divergence dictate the local conservation or divergence of the epigenetic modification. Before doing the experiment, we thought that as sequence conservation increases, the epigenetic conservation should increase. So what we started to do was to break down the human genome into 150 million 200 base pair segments. And on each segment, there is a conservation score based on FaceCom, which correlates to how fast do they change across the vertebrates. So that I would put the genomic segments to the right-hand side if they are pretty conserved, on the 17th spin if it's neutral, and on the first spin if it's somewhat positively selected or change fast in the human lineage. So now that we started to compare the sequence conservation level to the epigenome conservation level, to our surprise, not every epigenetic modification would have an increased conservation level as the sequence conservation level increases. To our further surprise, some of them, starting from neutral sequence to the negatively selected, as sequence conservation increases, the epigenome conservation increases. But starting from neutral to the positively, or the human genome, the parts of the human genome that change faster and faster, you have increasing conservation level of the epigenetic modifications on top of which, so that you start to have this basin type of bathtub shape of correlation between epigenetic conservation and epigenome conservation. So, and the next questions are how correlated are the interspecies change of the expression to the interspecies change of epigenetic modification? It turned out that we seem to see some correlation between the interspecies change of expression. Every dot now is a pair of orthologous genes between these 2 species. We couldn't find a general rule that would be able to correlate interspecies change of sequence to interspecies change of expression. It's not to say that we don't have individual cases that seems to correlate, where I can use sequence change to predict the expression change, but in a whole genome manner, out of about 13,000 orthologous genes, we couldn't find anything to correlate these 2. And the idea was that maybe epigenetic changes somehow correlate with transcription factor binding changes and then correlate with gene expression. That seemed to be the case, at least at an association level. So just to explore what we have seen in a different way, so this was the gene that we looked at. There are sequence conservation between these 2 species, between that 2 species, and between these 2 species. Epigenetic modification has strong signal here, and weak signal here, which correlates with strong expression, and a weak expression. So in general, we see more interspecies correlation of epigenome change to transcriptome change in general. So the next question we'll start to ask is, what are the types of epigenetic modifications that tend to co-appear with each other or don't like each other. So I'm looking at co-appearance scores between rows and columns. Some of them like to co-appear, which have some reasons, and some of them seem to do not like to co-appear with each other. Whenever they co-appear, they seem to be a lot more preferred to co-appear on orthologous sequences than just on any other piece of the genome, which that part of data I'm not showing. So from that, the group started to wonder if we can utilize the conservation of co-appearance of two epigenetic marks as a way to predict regulatory sequences. Again, we still do not forsake the conservation, but we just use a much lower level on the conservation level of the sequence. Now you add another criteria, which is the co-appearance of two epigenetic modifications on orthologous sequences. We wanted to ask these questions: when you have such things, are those things regulatory? Can you already start to tell what is the regulatory function of those sequences? How do I test this? We wanted to test it in a system where we change the cell behavior. So we use the differentiation system. During differentiation, suppose that I have some regulatory sequences, predicted regulatory sequences, that were predicted by co-appearance of conservation of co-appearance of two marks. Now that I predict this to be a regulatory sequence of some nearby genes, the way that we are going to test it is to perturb the cells. And during such a perturbance, you can have certain changes. You can only have these four types of changes. You lose either one of the two or you lose both. Thank you. And so after you have such changes, if this was indeed regulatory to this gene, then you can predict that if you lose the repressive mark, you would have enhanced expression level and so forth. That would be what you would predict to happen. So to test out this prediction, we generated a time course expression assay as well as epigenome modification assay at 4 time points during a defined differentiation process. Now we start to look at the following data. So for each modification, you have before, between, and after differentiation. Now we started to do some prediction. So there are co-appearance of two marks that co-appeared on some orthologous sequences and appeared in another species as well. Now you start to predict that this might be regulatory. What might be its regulatory function? Since the activation mark was kept and the repressive mark was lost, I might predict that this might be a poised promoter that would activate the transcription after differentiation, which appeared to be the case. This was a positive control because this pair was already worked out to be poised promoters. This doesn't appear to be true in only one case. This appears to be true throughout the genome for about 300 such poised enhancer promoters as we predict in this manner. Whenever you lose one, you go this way, you lose the other, you go the other way as you would predict. This doesn't only work for this one pair. This worked for 7 pairs of co-localized modifications. They seem to mark 5 types of regulatory sequences. Some types seem to be new, novel, poised promoter, or some other marks of other regulatory types. And so what we started to look at is to utilize the evolution as a way to look at the structure change across species. We might also, since now that the data is available to look at the dynamic change of the structure, that might give us some information beyond just binary code of the combination. So if I can remind you, the data that we are looking at is temporal data in a differentiation process. The example comes from here near this particular gene. So we wanted to look at beyond a combination of 2 kinds. So what we wanted to do now was clustering. We wanted to cluster genomic segments into clusters so that each cluster would have the same temporal change of the epigenetic modifications. So to model the temporal change, we used a hidden Markov model where the hidden state refers to the state of the regulatory code, and that would emit the temporal data. And that, if I could say in a nutshell, would start to give us this picture. So that for enhancers, we would have different combinatorial codes. You have these guys up, these guys down, which would correlate to certain changes in the output. We started to have different types of combinations that gives us, some gives us predictions of non-coding RNA change, some give us to messenger RNA change. So if I can summarize, we started to contribute to comparative epigenomics, not only genomics now. That was intended to use the interspecies epigenome comparison as a complement to genome comparison and use that to annotate the genomes. We started to have temporal data, started to have multi-species epigenome assays in comparable cell types. There are some efforts went in to develop some epigenome comparison browsers, which started to have some investigation of basic properties, including that the sequence change doesn't completely dictate epigenome change, including that there seems to be some correspondence between epigenome change and transcriptome change. And finally, conserved co-appearance of two marks seems to predict where the regulatory sequences are. So if genome is text, epigenome might be melody. If we can do genome comparison as comparing textbooks as the Rosetta Stone, maybe we can compare whole songs, including the text and the melody across species. And thank you very much for your attention.
For one question online, and others can ask him further offline. Any questions? Okay, we'll just break then for coffee, and thank all the speakers.